Okay, hi guys. This video is going to cover transcription, translation, and also mutations. So it's an overview of the whole process of protein synthesis and then also like what can go wrong with it. So here we go. All right, first of all, before we talk about the process of protein synthesis, we have to talk about the differences between DNA and RNA. So for the most part, DNA and RNA are really similar except for RNA is a lot smaller. So RNA contains only one gene. Um, it's single stranded, whereas DNA was double stranded. And then it also has a little bit different nitrogenous base makeup. So RNA has A, U, C, and G, and DNA has A, T, C, and G. So if I'm going to like write about that, in DNA, A pairs with T, and that also goes the other way, and C pairs with G. In RNA, the bases that are complementary are A and U, and still C and G. So that's one major difference. And then also the sugars are different. So RNA has ribose, which is why it is named ribonucleic acid, RNA. DNA has deoxyribose, which is why it's named deoxyribonucleic acid, or DNA. Also, there are 3 different types of RNA. So I'm going to put them over here. 3 types of RNA. There's mRNA, which is messenger. And that's the one that we're really going to talk most about. It's the one that contains one gene that will eventually be translated into a protein. There's tRNA, which is transfer. And its job is to transfer a specific amino acid onto a growing protein chain. And there is rRNA, which makes up ribosomes. So it's ribosomal. And we're not going to talk about that one too much, but ribosomes are made up of RNA and protein subunits. There you go. Also, one other really important thing is that when you have 3 bases of mRNA, it's called a codon. So codon is for mRNA. When you have 3 letters that are complementary to your codon, it's an anticodon. So in tRNA, it's an anticodon. So for example, if you had an mRNA code that was AUC, the tRNA anticodon would be UAG. This here is the codon. This here is the anticodon because they're complementary to one another. Okay, now let's talk about the process of transcription. All right, so you have learned this before in freshman bio, the process of transcription and translation. Together, transcription and translation make up protein synthesis, which is one of the most important things in biology that all living things do. It's actually called the central dogma of biology, transcription and translation together. Transcription is the step that takes DNA and converts it to RNA. So I'm going to put over here that transcription is DNA to RNA. And it could be any type of RNA. It could be M, could be T, it could be R. No matter what it is, it's made this way. How I like to teach this is that a transcription, or if you transcribe something, it means that you are keeping it in the same language, but you're just turning it into a different format. And that's what's happening here. So you're taking DNA, which is in the language of nucleic acids, and you're converting it into RNA, which is also in the language of acids. So it's like when you get a phone voicemail and you look at your phone in the voicemail section and it says like transcription not available, which means it couldn't like type it out for you. It's still in English. It's just changing it from words spoken to words written. And that's what transcription is. It's changing it from DNA, nucleic acids, to RNA, nucleic acids. So it's the same language. Okay, then also on the slide are the different types of RNA. I already wrote them on the slide above, so I'm not going to write them again. You can rewind me if you need to. The process of transcription begins at the promoter and ends at the terminator. This is in prokaryotic organisms. In eukaryotic organisms, it's a little bit different. We have proteins called transcription factors that bind to the DNA to start transcription and to signal that it's supposed to start. For bacteria, they don't have that. They have a specific region of the DNA code that signals this is the start point for transcription, and they have a certain end sequence, like a certain sequence of letters, A's, T's, C's, and G's, that signals this is the end. So for eukaryotes, we can start transcription at any point, just depending on where the transcription factors bind. For prokaryotes, it will always start at the exact same spot. In eukaryotes, we also have like a sequence at the end of each of our genes that signals, okay, this is the end of the gene, and then proteins will cut that RNA off at the end. That's just one small difference. There are more that I'm going to talk about in a minute. Basically, when you do this, you use a whole bunch of enzymes to make an RNA version of your DNA. You're only going to make one gene at a time for this because you're only trying to make one protein. So it's a pretty small segment of RNA and a small segment of your DNA that's used for this. Just like in DNA replication, there are tons of enzymes that need to be used to make this happen. One of them is called RNA polymerase, which remember, we had DNA polymerase that replicated the DNA. RNA polymerase is going to add new mRNA nucleotides into the growing mRNA or tRNA or whatever RNA you're making. And then this always happens in the 5' to 3' direction, the production. So the DNA is read from 3' to 5', just like the leading strand would have been in replication. So it doesn't have to be copied backwards in fragments. It's always copied forward. So the 3' to 5' strand is the template strand. And it always is the template strand, so that the mRNA always ends up being 5' to 3'. All right. Here's a picture of this happening. So you have the DNA that's opening up. RNA polymerase is able to unzip the DNA and also add in the new RNA nucleotides. The RNA nucleotides are red. You have the template strand on the bottom, which is running 3' to 5'. And then you have your newly made RNA that is 5' to 3'. This is corresponding to the end of this RNA. So down on the bottom, you have, this was your template strand, the dark blue. And here's your template strand again. So this over here is the start of the mRNA, and then this is the end. So you're running 5' to 3'. And then you can see which letters are complementary, of course, remembering that A's and U's do not pair, or they do pair together in mRNA instead of A's and T's. So here's a T on the DNA, it's pairing with an A. Here's an A on the DNA, it's pairing with a U, because there are no T RNA nucleotides, no red T's. All right, so then for eukaryotes, once the process of transcription is finished, there can be modification of the RNA, and there is modification of the RNA. So once transcription is immediately finished, we call the RNA pre-RNA because it's not ready yet to make a protein. And what it has is like all of these extra segments of DNA that don't actually need to exist to create the protein, and they shouldn't exist. So they're going to get cut out. First, though, we're going to have end modification. Slide got a little weird. The 5' end, which remember the 5' end is the beginning, is going to get a cap that is guanines or Gs. Um, and that's going to just like prevent any damage from happening to the beginning of the gene. It's literally called a cap. Um, and it gets added once there's been about 20 to 40 nucleotides passed. It will get added to the front. This is going on the front side. And then on the end, there's a tail that gets added, and it's a bunch of adenines that basically prevent the RNA from getting broken down before it gets to the ribosome. The tail is really interesting because as soon as the mRNA leaves the nucleus, what will happen is enzymes that are in the cytoplasm will start to degrade that RNA. So the poly A tail, which is on the end, is really important for making sure that the RNA is preserved long enough to actually produce the right protein. RNAs that need to be used more than one time will typically have a longer tail because they need to last longer in the cell. And then RNAs that just need to be used very quickly and don't need to make a bunch of copies of that protein will have a shorter A tail because the cell doesn't need them for as long and they can get broken down. So both the cap and the tail help the RNA make it to the ribosome to actually be produced into a protein. The other thing that happens is splicing, and that is when you cut out the unnecessary regions of the RNA. There's two names for these things, the different regions. They are called introns and exons. Introns are the ones that get cut out, which for some people feels like a little bit backwards, but that's the way it is. So introns are removed. Exons exit the nucleus. And that's how I usually remember that exons are the coding region, so they're the ones that have to leave the nucleus. This is done using something called small nuclear ribonucleoproteins, which we abbreviate as SNRPs. That's how you pronounce it. Here it is. In class, when I do this lecture, usually I have like a little SNRP that has scissors that sits on the desk. But sad day, it's not here. Anyways, so the SNRPs join with some other protein complexes to form something called a spliceosome. And the spliceosome is responsible for two things. One, cutting out the introns, and two, sticking the exons together. So basically like taking this part out, these attach. And then you have RNA that's ready to leave the nucleus. Here's a picture of what that RNA looks like. So you have the 5' cap on the start end, and then you have your poly A tail on the end. In the middle is the actual protein coding segment. So this right here is the part that actually will make the protein. It's flanked on both sides by a start codon and a stop codon, and those are the signals to the ribosome like, okay, we're starting, or okay, we're ending. On either side is a UTR, which is called an untranslated region. That's what that stands for. Take a note of that. Untranslated region. It's just a segment of the RNA that's not actually going to be part of the protein. And then finally, at the end, you have the poly A tail, which could be over 100 A's, or it could be just a few. And that's your finished mRNA. This is only in eukaryotes that this modification is able to happen. In prokaryotes, there is no modification at all of the mRNA. And the process of transcription and translation is basically almost simultaneous. Here's another picture that just shows what the mRNA looks like when there is the introns. Remember, the introns will be removed, so your mRNA ends up getting a lot shorter. The exons are stuck together to create the coding segment and also to create the untranslated regions that are kind of like just on either side of the gene. And then here's what a spliceosome looks like. So you have your SNRPs over there on the left and then some other proteins. They combine together into a bigger unit. and they're responsible for acting like little scissors, chopping out the introns and then sticking together the exons. And then the introns are just degraded by the cell using enzymes, and the mRNA is allowed to leave the nucleus. All right. So then it's time for the process of translation. So, like I talked about transcription being like you're keeping it in the same language, you're going from nucleic acid to nucleic acid. In translation, you are translating it into a different language, just like you would be if you were like using Google Translate. So, what happens here? Translation is DNA, I'm sorry, not DNA, RNA to protein. The language of RNA is nucleic acid. The language of protein is amino acids. So that means that you're taking the message and you're converting it into something new, which is the protein. And that's what translation is, and that's why it's called that. So basically what happens is your mRNA that's been processed and like fixed up and the introns have been removed leaves the nucleus through a nuclear pore, because all of this so far has been happening in the nucleus. That's where the DNA stays and it never leaves. Now this mRNA is leaving. So it goes through the cytoplasm to the ribosome, and it will be read from beginning to end, 5' to 3', starting with that cap and ending with the A tail. Ribosomes themselves have two parts. They're a larger subunit and a smaller subunit. Really what they look like is this sort of deal where this is the larger subunit and then underneath is the smaller subunit. Looks like a Krabby Patty. And then what happens is the mRNA will thread through here and be read as it threads through. This blue thing is the ribosome. This green thing is the mRNA. And then, so what happens is as they interact, there are things called initiators, which attach to the start sequence. The start sequence always codes for the amino acid methionine, and it's always the same 3 letters. It's AUG on the mRNA. Oops. And it always is that. So then what happens is tRNAs carry amino acids to the ribosomes to be assembled. They know which amino acid to bring because each tRNA always corresponds to that same amino acid. So for example, if my mRNA had AUG, then a complementary tRNA would be UAC. That UAC always carries methionine. And that's how it keeps the right amino acids coming into the protein. This will continue all the way until a stop codon is reached. Stop codons code for the protein to be terminated and released to the cell. And then you have a newly synthesized protein released. There are a few options of what it could be doing or where it could be going. If it was produced by a ribosome that was attached to the endoplasmic reticulum, a bound ribosome, then that protein will be like packaged and shipped out of the cell. Maybe it's a hormone or something like that. If it was produced by a free ribosome, it will be used within the cell. So maybe it's an enzyme that the cell needs or a membrane protein or something like that. And then the proteins basically make you who you are. Like all your traits are just different proteins being expressed compared to other people. So it's really, this is like why you are who you are and why you look how you look and all of that, which is cool. Okay, here's some pictures of translation, how it works. The brown is the ribosome. It has the larger subunit on the top and the smaller subunit on the bottom. And then, like I said, the mRNA is threading through. The 5' end is the beginning and the 3' is the end. This is the end. For each codon, there is an anticodon on the like light blue tRNAs that is complementary. The codon will only bind to a complementary anticodon, and the anticodon will only be carrying one specific amino acid. So what's cool about this is once the tRNA drops off its amino acid to the chain, it will leave to the cytoplasm and go find that same amino acid to pick up again. Where those amino acids come from is from your food. So that's why you have to eat proteins, because the proteins that you eat are broken down to amino acids and then used to create new proteins that are you, which is really amazing. So in some ways, like, you are what you eat, because the pieces of your food get used to make you. Recycling. Okay, here is a codon chart. You have seen this before, but you might have forgot. This works to tell you, based on your mRNA, what tRNA will result and what will bring the correct amino acid. So you read this basically by going on the left side and then the top and then the third and looking for the place where those interact together. So let's say that I had AUG. Let's just use AUG. So A is here, U is here, and G is here. So I'm looking for the place where all three of those interact. Of course, it would be AUG. The amino acid that goes with that is methionine. So that's how I would know, okay, methionine is the amino acid that goes in that spot. You can see there's 3 stop codons up at the top. Those are codons that would not trigger an amino acid to be added. They would trigger the protein to be cut. And they're really important for making sure the protein gets cut at the right time, because if you don't have a stop codon, you start going into your untranslated region, which isn't part of the protein. And if you put those in, it's going to change how the protein folds and change the function of the protein. So, yeah, cool. Here's the whole process all together. You can see up at the top, there's the DNA. The DNA is going to use the 3' to 5' end to create the mRNA. The mRNA has U's instead of T's. Then the mRNA leaves the nucleus. It goes to a ribosome. Each codon corresponds to a specific amino acid, and that produces your functional protein. There are 20 amino acids that exist. 20 total amino acids. You will see up at the chart, I'll go back real quick, that there are multiple codons that can code for the same amino acid, and that is because there are only 20 amino acids, and there are many other options for codons. So that's kind of nice, because if you get a mutation, a lot of times, especially in the 3rd spot of the codon, you can end up coding for the same amino acid anyways. Like if you look at this one here for arginine, no matter what your 3rd letter is, if you have CG, you're going to code for arginine. And that means that if you had a mutation, it would have no effect on the function of the protein, because it would bring the same amino acid anyways. So it would be totally fine, which is really nice. That's called wobble. Wobble means 3rd base of codon can change and still code for same amino acid. And it's not always true for all of them, but it is true for many of them. So like I said, sometimes it doesn't work, but for many amino acids, it is the case, which is nice. So in summary, just comparison for prokaryotes, I'm going to talk more about them at the end of the week, but in bacteria, transcription and translation basically happen at the same time. There is no modification of mRNA after transcription. So basically, like, what you get in the DNA is exactly what you get in the RNA. And of course, RNA doesn't have to leave the nucleus, because there is no nucleus in prokaryotes. So everything is happening in the cytoplasm. Here's the comparison of that. So you have the eukaryotic cell on the right, which is what I've been talking about the whole time, with the nucleus and the DNA, pre-mRNA, which gets processed into a functional RNA, leaves the nucleus and does translation. In a bacteria, it's happening a lot faster because everything's in the same spot, and there is no processing after transcription. Okay, last thing is mutations. So mutations are changes in the DNA sequence. It's really important that they are changes in the DNA sequence and not in the RNA sequence. So Mutations happen during mitosis or during interphase of mitosis during S phase, during DNA replication. So occur randomly during S phase. For the most part, your cells are really good at catching mutations and making sure that they don't get passed on to other cells or fixing them. But sometimes they don't, and that can lead eventually to cancer, or it can have like no effect, like I said before, or they can even be beneficial. There are a few different types of mutations. One of them is called a point mutation. Point mutation means literally just one base was switched. A lot of times they're substitutions, which means like you traded a T with an A, and that's all it can take to cause a disorder like sickle cell anemia is caused by a substitution in one letter. And it's just about the location where it is, coding for a different amino acid that changes the folding of the hemoglobin protein in that case. Substitutions cause missense mutations, which this is right here, that the new base codes for a different amino acid and not the correct one. It is possible that it could have no effect at all, and those are called null mutations. I'll just say over here, null equals no effect on organism. So you could substitute, like I said, in wobble, the third base of the codon and still code for the same amino acid. And it's still a mutation, it's just a null mutation, which means it didn't matter. And then there's also a different type of point mutations called frame shift mutations. And they're either insertions or deletions. And they're usually worse because what happens here is you're either adding or deleting a base. I like to demonstrate this using actually like an English sentence that's all 3-letter words. So the cat ate the rat. In this situation, each of these 3-letter words is a codon. So then let's say that we're gonna add another, we're gonna have an insertion. We're gonna insert another E. We're gonna insert an E here. Now, because things have to be read in groups of 3, it's gonna continue to be read in groups of 3. So you can see here that this makes literally no sense at all. It's a much, much bigger change than if we were to have, I'll do a substitution. So in this case, we had a substitution here. You can see the change is a lot smaller. So in the frame shift mutation, which is the one at the top, the amino acids that follow and the codons that follow, none of them are what they should have been. None of them are right. With the substitution, most of them are right, but one of them is not. And that one could be a null mutation or it could have an effect, but it's a lot more likely that your codons in a frame shift are going to be really, really wrong. So I'm just going to label that this is a frame shift. This is a substitution. Both of these are point mutations, which means that just one letter, one base is the cause of the mutation, as opposed to like a translocation where you have a chunk of the chromosome being moved, like a really big deletion where you have like a chunk of the chromosome being deleted. Those are bigger deals. These are smaller mutations called point mutations. All right. Oh, the other thing that I did want to mention here is sometimes you can have a point mutation that causes a stop codon to be coded for too early. Those are called truncation mutations, where you have a protein then that's like much, much, much shorter than it should be and non-functional usually. Okay, so mutations, of course, can be random, like totally random, or they can be triggered by things called mutagens, which are agents that cause the DNA molecule to become damaged. Um, One example of that is UV radiation. There are tons of things that are have been like deemed mutagens. Carcinogens are chemicals that like lead to cancer, and a lot of those are also mutagens. Um, UV radiation causes like the base pairing to go wrong in DNA replication. Sometimes you can have like double-stranded breaks as a result of this, where literally like the DNA ladder like breaks in half. Those are really hard for the cell to repair. Um, they're just chemicals that trigger mutations at a higher rate than normal smoking. A lot of the chemicals in smoking or in cigarettes. And then, of course, I've said this, but not all mutations are always bad. It just depends on what amino acid and what protein gets affected, and it's usually totally random where the mutation lands. Most of the time, it actually has no effect, but it can, of course, be negative or it can be positive. And an example of it being positive is that it increases the genetic variation of the population, which is how we got all of the living things that exist today is through mutations that built up over time and were passed down through generations and like caused the differences that exist between species. So mutations are actually a really important driver of evolution and really important in contributing genetic variation to a population, which helps them survive like catastrophic events. All right, here's some pictures. This is an example of the sickle cell mutation. So you have whenever you see wild type, that's like the normal. So on the left, you have the normal hemoglobin DNA, and then on the right side, you have the mutant. So you can see it's just one substitution that causes this. The substitution happens in the DNA because that's where mutations happen. And eventually that triggers the wrong amino acid to be added to the hemoglobin protein. That affects the way the hemoglobin protein folds so that it doesn't fold into a round globular shape. It folds into a long, like sickly shape. So there you go. One letter causes that. Here's just some other examples of frame shifts and all of that. Um, you can see here the different like effects that occur. Over here, you have a silent mutation. That's another way to say no effect. And then you have your missense, which means the wrong amino acid like gets added in. You have a truncation down below that shows a stop. That's nonsense. This is like on too early. Truncation. Missense is wrong amino acid. And then you have your frame shift, two examples of a frame shift. And then also down here is an interesting situation where you have a deletion of 3 nucleotides. So you deleted a whole codon. That's like in our sentence about the cat. It's like deleting a whole word. So it's still wrong, but it doesn't cause the frame shift because it's even 3 that get released or deleted or added. So it's going to just either delete a whole amino acid or it's going to add a whole amino acid in. Here's just one more example of a silent mutation, which means that it has no effect on the organism. This is an example of wobble. So you can see that the mutation happened in the third base spot and still coded for a glycine like it was supposed to. So I'm going to label this as wobble, which is nice because it would have no effect on the organism. All right. Here's a zoomed in missense mutation where it shows the different amino acid being coded for. And now it's a serine instead of a glycine. This one is a mutation in the first spot of the codon. So you can see like if you have a mutation earlier on in the codon, you're going to have more effects on the amino acid that gets coded for. And then here's a truncation where you have the stop codon being coded for too early because of the change in the DNA. So you have UAG, which codes for stop, so the protein's going to be cut off right at the beginning. This is going to be nonsense because it's literally not going to be a protein. It's just one amino acid floating around. So it has no function at all. Doesn't even really exist. So that is a early stop codon truncation nonsense mutation. And that's it. Hooray. Okay, so hopefully you enjoyed this. And I'll see you soon. Good job.